Hi guys! Our topic is about epigenetics. My part are relationship of gene expression and cell differentiation, and the one is control of eukaryotic in gene expression. So everything in our body is made up of cells. Your hair, fingernails, bone, muscles, organ, and even blood. So all these cells contains of DNA or genetic materials. So DNA is what makes you unique individual. So how is it possible that the cells contain same information yet look and act so different from each other? So cell differentiation is how genetic embryonic cells become specialized cells. This occurs during the process of gene expression. So gene expression is the process of combination of genes that turn on or off, express or repress. And also it indicates how a cell function. So in general, the relationship of cell differentiation and gene expression in the process of cell differentiation, which is the cells dramatically change each shape, size, metabolic activities, and responsiveness to the signals and membrane potential. This occurs because of highly control of modification in gene expression. So next is the control of eukaryotic gene expression. So in prokaryotic cells or bacterial cells, the operon is used to generate gene expression. So how about in eukaryotic cells? So I will discuss the control of eukaryotic gene expression and how it is regulate, because eukaryotic cells is more complex than eukaryotic. In any eukaryotic genes, we have those segments of DNA, those segments that code for a polypeptide and those segments that do not code for a polypeptide. So in this diagram, these are the exons. The exon 1, exon 2, exon 3. These segments are code for a polypeptide. And the next one is the intron 1, intron 2, which is they do not code for a polypeptide or anything useful. Eventually, in the process of RNA replacing, these segments or the introns are actually removed from that mRNA molecule. So next is the transcription start site. So in this diagram, this is the transcription start site or the start site. So transcription start site is this is where the transcription actually begin. So the special protein complex that consists of 12 protein is known as the RNA polymerase II that binds onto the transcription start site. So this is actually the process of transcription. So the next segment is the promoter. Promoter can broken down into a core promoter and upstream promoter. So core promoter is located in the left side of the start site. This is the core promoter. And the most common type of core promoter is the Tata box. So why is it called Tata box? Because the sequence of nucleotide is TATA. So what is special about Tata box? Tata box, where all the transcription factor and regulatory protein bind to and form a complex that consists of 50 proteins, 50 different types of proteins. And it also call other proteins that interact with a protein found in the start site. So in that, initiate the RNA transcription. So as I've said a while ago, the promoter can broken down into a core promoter and the upstream promoter. As we can see that the promoter is the majority of the protein. Then the remaining promoter is called the upstream promoter. Upstream promoter is also allowed binding different types regulatory protein. So unlike the core promoter, which is do not change one eukaryotic gene to another, the upstream promoter can vary different types of eukaryotic genes. So, the last segments or the final segments is the enhancer. So, based on our first diagram, the enhancer is located here. So, the enhancer is further away from our genes. And also, enhancer bind special transcription factor. Now, when transcription factor, this is the transcription factor, when the transcription factor bind into the enhancer will loop around and bind onto the protein complex down to the promoter. So it can be upstream and it can be downstream. And now, the enhancer can actually increase the rate of transcription once the binding takes place. And that stimulates the transcription of that particular genes. So, this one method by which eukaryotic cells actually regulates. Thank you.